Thank you for the very nice introduction and for the lovely invitation to be here. So yeah, I'm going to just talk a bit about some of the research actually we're doing in my lab, but I'm hopefully going to put it into enough context and explain enough of what we're doing and why we're doing it that it will make sense to this varied audience. I understand there's all kinds of backgrounds and interests here, so I'll hopefully explain myself well. But I am happy if at any time I am confusing, just wave at me and you don't have to wait till the end to questions. I'd rather not have everyone sit confused through the majority of the talk than sit silent. So please do interrupt me if I do get confusing. But if we're talking about, so I'm going to be talking about things that we think have happened at the really early on in vertebrate evolution. But here on this particular collage of pictures here, we have a tiny sample of the amazing biological diversity that exists on this planet. So, of course, we have some vertebrates that are all kind of quite strikingly different from each other. But we've got these other things, the C. elegans worm, octopus, which are kind of amazing, jellyfish, which have eyes but not a brain, so I don't know what they do with the information. And then some other extraordinary eyes over here. We've got some plant life, and then of course there's implied in these pictures, of course, there's tons of bacterial life as well, and probably some viruses that we can't even see. So there's an extraordinary diversity of life on this planet. And so how do we have all of this diversity? How did this one grow into a human, and this one grow into a fish, and this one grew into a frog? Well, I suppose the trite answer is because this one had human DNA, and this one had frog DNA, and this one had parrot DNA. But so in a sense, we know that our DNA really makes us what we are. So we all here grew into humans because we grew from a fertilized egg that contained human DNA. It has the instructions to make us what we are. And so all of these different forms of life, be they different animals or even plants and other weird things, they've got different DNA sequences, and often they have different genes from each other. So there's also, it's not just that the sequence changes a little bit in terms of a letter changing to another letter, because we can write DNA down as letters, but also you get new genes. So sometimes we have totally new genes in these different animals and plants and everything that are doing different things. So sometimes we get these new functions, these new adaptations, these new behaviors because of new genes. So then we need to ask ourselves, where do these new genes come from? Because genes are complicated things, and it's not a lie that we don't exactly know what genes are, and there's kind of a few ways that sentence is true. One is that we don't really have one definition of a gene that works all the time for everything. So some of the times you can say, well, I know it when I see it. And so there's things that we know are genes. But one working definition, one operational definition of a gene is a contiguous piece of DNA on a chromosome that codes for a protein. So that doesn't work all the time as a definition. There are genes that don't code for proteins, and there are other things, but that's a pretty good definition. But even by that definition, we don't even know them all. In the human genome that we've had available in draft form since 2001, and it was more complete a couple of years later, we're still not 100% sure how many genes we have. And we're a very narcissistic species, and we spend a lot of our time analyzing our own genome and our own species, and we still don't know everything about it. But even still, when we look at a gene, if we think about this gene that's a contiguous piece of DNA that codes for a protein, there are lots of things involved in that. There are bits of that sequence that need to be cut out exactly precisely in order for that to be read correctly as a protein coding sequence. There are signals that are just beside the gene that tell it when and where to turn on. And there's all of these kind of things. So it has to have the right code, it has to have the right signals for cutting the bits out, it has to have the right signals for turning on in the right place. And so it's really, really complicated. So what we see is most of the time, nearly all of the time, evolution takes a shortcut because where new genes come from is from other genes. So what we see is that genes tend to get copied. So then you have an actual physical copy, an extra copy of a gene in your genome, And then after that, it changes. So what we see is, if you imagine a scenario, this obviously a piece of DNA, and just this blue box here is supposed to be representing a gene. And if you imagine you've got a situation where you've got a gene, then you just literally have 2 copies of that gene. And initially, those would be 2 identical copies. So it's your DNA sequence, your genome sequence just gets longer by a little bit when this happens. This kind of mutation event happens all the time. This variation exists in this room, I'm sure of it. And so you get extra copies of things. And initially, they're identical. But over time, they can change a bit. And these changes can be quite powerful. And I'll give you a few well-known, well-studied examples. So one is to do with how we can see all the colors we can see. So this obviously is a drawing of an eye with the lens at the front. And this pinky red bit at the back is supposed to be the retina. So if we were to take a zoomed in look at this bit of retina, then what you would see is there are a few different types of cell in your retina. So there are rod cells and cone cells, very artfully depicted here. They are not so obviously, they're not so obviously rod-shaped and cone-shaped, but they are approximately, which is how they got their names. And these different cells in the retina, one of the things that distinguishes them is they have present in them different versions of an opsin. So an opsin is a light-sensitive pigment that's produced, it's a protein produced by a gene. So in the rod cells, there's rhodopsin, which you might say is responsible for black and white vision, approximately speaking. And then in these cone cells, there's 3 different kinds. So there's either short-wave sensitive, medium-wave sensitive, or long-wave sensitive opsin. And so these are approximately in the blue, green, and red areas of the spectrum. So when we look at the sequences of these genes, part of the reason they're all called opsins is not only because they do the same thing, but when you look at the sequence of the gene, you can clearly see that they are very, very similar. There's small differences in the sequence of the gene, which make small differences in the protein, which make these small differences in what wavelength of light excites that protein and sends a signal. So these are copies of genes. These were initially identical copies that changed over time to have these slightly different functions, which we have biologically have harnessed to great effect because we can see thousands of colors because of the combinations of these three different opsins. Another example where gene duplication has been really important in terms of getting new genes is in the globins. So you've probably heard of hemoglobin before in your blood carrying oxygen. There's also myoglobin, which is in your muscle tissue holding oxygen there. And hemoglobin, in fact, is made up of alpha and beta globin. And then myoglobin is just one bit on its own. So those are all copies of genes that were initially identical and then changed a bit. And so in this case, they've changed subtly in one respect because we still see at one fundamental level they're doing the same thing. They're binding oxygen and carrying it, but they've specialized for different parts of your body. But even within hemoglobin, there's a very interesting specialization, I think, which is a specialization for different stages in development. So this x-axis here is tracking, this is fetal development until birth, and then a baby up until 48 weeks. So in a 48-week-old baby, just like in adults, the hemoglobin is made up of alpha and beta globin. So if you look in a 48-week-old baby, you'll see that their hemoglobin is all alpha and beta globin together. But if you were to look at the blood in utero of a developing fetus here, you can see that, so there's some things going on here early on. There's this globin that's gone quickly and is taken over by alpha. But here in the womb, instead of beta globin, which is quite low, in the womb, a baby has a lambda globin. So this is a copy of the beta globin gene that's changed a bit. And the way it has changed is to be more attractive to oxygen. So if you imagine what's happening with the oxygen transfer in mammals, so animals that lay eggs, they leave their egg there and the oxygen is going to diffuse in through the cell. But in mammals, in placental mammals, the mother is doing all the breathing, the oxygen's going into her blood, and the oxygen has to be exchanged over to the developing fetus without the blood mixing, because there might be a different blood group and that would cause clotting and all of these things. So in the placenta, the oxygen has to be transferred without exchanging any blood. And so the fetal hemoglobin has a stronger pull on the oxygen, so manages to steal, in a sense, the oxygen from the mother's blood. So it's by the chemistry of this protein that oxygen transfer can occur in the placenta, which is just kind of wonderful. So, but anyway, this is just another example of how you get new functions. So even though you start out with an identical copy, you can get new things happening. But I have digressed, in a sense, from the vertebrates. So that, if we get back to the idea of looking at vertebrates, so just here are some vertebrates. So this human is slightly occluded by this wonderful elephant shark, but this particular human here is my collaborator who sequenced this genome. So he gets in there just about. But we have this interesting diversity of vertebrates, and I've only just picked a tiny, tiny sample here. I think the slide I had earlier had some almost more striking examples, but one of my favorite dinosaurs here that we can see this time of year as well, and some other peculiar ones. So if we want to, one of the things we want to do, or I'd like to do anyway, is try and understand what was happening at the base of the vertebrate tree. So at that way back when, approximately 500 million years ago, when before all of these species radiated out and accumulated all of this diversity. So in order to try and understand what happened to create the huge diversity within the vertebrates. And one framework for doing this is thinking of them in terms of their phylogenetic tree, so their species tree, which is like a kind of family tree. And then you can make inferences about the different steps along the way in terms of the common ancestor. So if we compare the species in terms of this phylogenetic tree, we can, in a sense, start reconstructing, imaginatively reconstructing the ancestor. So what do we mean by this? Well, if we were to take these species that we were just looking at there, for example, we see that the bird and the snake are, of the ones here, those are each other's closest relatives. So if we wanted to make some inferences about this ancestor that would have existed back in time here, so we're going back in time as we go back here, then we might say, well, that ancestor, one of the things we would say about it is that it had scaly skin. So birds have scales, you just don't necessarily notice it. If you look at their legs, you see it, and their feathers are actually modified scales. But we could say this thing had scaly skin. We can make inferences like that. And if we ask about the ancestor of all of these ones, so we throw in the mammals there, then we say, well, one of the things about this ancestor is it had 4 legs or 4 limbs, so it's a tetrapod. So I am aware that putting in a snake as an example here is very funny because we don't think of snakes as tetrapods, but they lost their limbs. So the ancestors of snakes used to have them. And you can see this actually if you look at what are called basal snakes. So the boa and the python. Am I getting that right? The python, I think so. Definitely the boa, because I decided to remember it as feather boa. But they are called basal snakes, and you don't see them protruding out of their skin, but if you look at their skeleton, you can see vestigial limbs. They're still there, haven't fully disappeared. So those have lost them. But if we go back further in time and we ask, well, if we throw in these bony fish here, we go, what would this ancestor look like? Well, all of these things have hard bones. So we could say this was a bony ancestor of all of these vertebrates. This elephant shark here only has cartilage like other sharks. So this comes in out here, and this ancestor here had cartilage, probably had a cartilage skeleton. And this thing here is a lamprey, and we're going to talk more about that in a little bit later. I don't know if anybody knows anything about a lamprey, but it's a quite cool, quite peculiar thing. But if we put this one in, we get to the base of the tree here, and things we're missing are things like there's no bones, there's no hard bones, and there's not even a jaw. So these are sometimes called jawless fish. So we can make these inferences by doing this comparison, and we can say it steps along the way, kind of where things originated. So we can do this for the morphological traits, but we can do exactly the same thing for genes, and we can start saying what ancestral genes look like. So here's just a bit of DNA sequence. So this is real DNA sequence. It happens to be a small snippet of a gene that codes for a developmental gene that is involved in forelimb development, frontal limb development. And here we've got a few examples of animals that have very different limbs. But if you look, you can see quickly anyway that the sequence is really similar. You can recognize, it's quite easy to recognize related genes across these species. And you can see there's some changes. So we have a T here in human where we have a C everywhere else. So if you were to say, well, what happened on this tree? The simplest explanation of what happened is that there was a C in the ancestor and it remained unchanged everywhere except just one little change happened on this branch to humans. So we can infer that the ancestral sequence here had a C in this position and actually a C in that position and things like that. We can infer what the sequence looked like. And as we understand more and more about sequences, we can even infer some of the traits from knowing the sequences. So, but if we want to understand, because I'm interested in knowing what happened really at the origins of vertebrates in order to try and understand what was special or what might have been special about the evolution just in that period that gave rise to this huge diversity within vertebrates, we need to find an even deeper relationship. We need to go just outside the vertebrates. And we find ourselves with this lovely guy. So this thing is commonly called a sea squirt. The scientific name is Siona intestinalis. So I don't know Latin. I can see this as intestines, but I'm reliably informed that this translates as column. So this is what they look like. And so the name is a column of intestines, which is kind of what they look like. So these things, you would not feel that if I just showed you this picture as I'm doing now, I don't think you'd guess that these are closely related to vertebrates. But these are the closest animal to vertebrates that is not itself a vertebrate. So this is a chordate. It has basically some kind of thing that you might call a spinal cord in us. So it has that structure, but doesn't have any kinds of bones. So this is the next thing just outside vertebrates. So when you put it on the tree, it comes out just here. And so when you compare this to these other ones, you can start asking, what was this ancestor looking like? And so when we look at that and we look at the genes, we want to ask the question, can we find any patterns? Can we see any patterns when we're comparing the genome of this sea squirt, this just our next cousin outside the vertebrates, and with all of the vertebrate sequences that we can look at? And are there any patterns that we can discern, and can we learn anything from that? And the first pattern, well, one obvious pattern that we can see is when we look in the sea squirt, in the Siona genome, and if these little balls are just genes, we approximately, as a first approximation, we see a roughly 1-to-4 relationship. So there'd be one gene in this Siona, and there's 4 copies of it in, for example, this human, who I haven't picked at random. He's a very influential person in the field, Sumo Ono. But so these are duplicate copies. So I was mentioning the gene duplication. But so there's lots of duplicate copies of genes. So where the Siona has 1, we have multiple copies, but it's not just a random number of multiples. It doesn't look like there was just lots of scattering of duplications going on. There's a strong trend that there's kind of this 1-to-4 ratio, or close to 4 ratio. And so something unusual happened here, and this unusual thing is considered to be somewhat very important in this amazing diversity we see in vertebrates. So it's considered to be an event or a series of events that created a genetic opportunity for all of the subsequent evolution to take place. So what do we think happened? So how can we explain this 1-to-4 ratio? So what made this happen? And of many genes, not all of them, but it's a strong ratio. So what we think happened is, instead of just gene duplication, instead of just genes getting copied, that the whole genome, so all of the DNA got copied. So in this cartoon version here, this is a simple genome that contains these 5 genes. And if this whole thing got duplicated, then you'd have just 2 identical copies of this, like that. And if this happened again, you'd have 4. So you can go from 1 to 4 by these 2 rounds of the whole genome getting duplicated. So this is what we think happened at the base of the vertebrate tree. And it created these huge numbers of genes in our genome, which seem to have perhaps created the conditions conducive to all this amazing evolution in terms of morphological and behavioral changes in vertebrates. So this, it isn't this perfect, so we don't just have this exact 1-to-4 ratio across everything. So one thing that happens is that many of these genes can subsequently get lost. So you get something that happens that disables a gene, or it just physically gets cut out of the chromosome. So when you look, you see this pattern a bit more like this. So only some of the genes are actually there kept as duplicate copies, but you see some examples that are, and this becomes important later, you'll see. But so this is the same sequence that I showed you before, to reiterate the point that oftentimes it's very easy to see that these are the same gene in the different species. You can see quite simply by looking at the DNA sequence, these really do look the same, and this is just a very small bit of sequence. This is taken out of a much, much longer string of sequence, where this is the pattern that you see across the whole thing. It's very obvious when you're reading these sequences that despite these changes, these really are the same gene in the different species. But it isn't always so easy to identify the genes like that. And so you can do this to identify across species. You can also do it to identify those copies of each other. So you see this when you're comparing the globins, and you see this when you're comparing the opsins. But it isn't always that easy, which means we can't always recognize these patterns and these deep patterns coming from the origin of the vertebrates. So one reason is that some of the genes they just don't have that similarity anymore in terms of their sequence, maybe because they have evolved very, very quickly. Lots and lots of changes have occurred, and it's very hard to tell the difference between that and just a random sequence anymore. You just lose information, lose signal. Or sometimes that can be not only because the gene is evolving quickly, some genes evolve quicker than others, also because the gene is short. And so once you start accumulating changes, it becomes much harder. So sometimes these relationships are really hard to detect. So we want to do something more to try and understand them. And so, for example, one way that we might be able to detect these difficult to detect relationships would be by their shared descent from a single bit of chromosome in an ancestral species. So if we imagine this piece of chromosome is in that ancient vertebrate genome, and it's been passed down through all the many, many, many generations over 500 million years. It's gone through that 2 rounds of duplication thing, so there's a 1-to-4 pattern. But when we look at this compared to that, compared to that, compared to that, it's very hard to see the relationships. So if we're looking in the human genome, it's very hard to see that pattern of relationship. But if we were able to say they all descended from this little segment on the ancestral genome, we'd be able to recognize now that they are related, if that makes sense. So this is what we can have these cryptic relationships where it's hard to know what's going on, and we'd like to know because it's important for many reasons, and we'd like to say, well, if we can solve this problem somehow, if we can just see if we can link them by their relationship with this, then maybe we can recognize these relationships that we couldn't see before. So if we knew that structure of that ancestral genome, then we could try that 4-onto-1 type mapping of the modern onto the ancient ancestral genome. So can we do this? Can we infer the structure of this ancestral genome? So this is the same This is the same group of species that I showed you before. And this is the lamprey that I promised to tell you more about. And so this is a funny little thing, or big thing, or scary thing. So when you say it's jawless, this is why it's jawless. So it doesn't have a jaw like we have. It's just got this weird circular sucker mouth. So you can see this because this one's in an aquarium. So it's up against the glass. And so you have this terrifying mouth that looks just like a little array of teeth. So this animal is a vertebrate. It doesn't have a jaw and it doesn't have hard bones. It's only got cartilage, and it's one of the earliest, one of the only examples of that early branching off in the vertebrate tree. It's weird not only to our eyes because of this, but it also has strange things that happen in terms of its genome, which I'll just tell you as a little aside. It's not relevant to what I'm going to be telling you later. I just find it fascinating. So in terms of your DNA that you have, you inherited that DNA from your parents, of course. And one thing you might have already heard is that that DNA is the same in all cells of your body. So if you want to identify somebody in terms of forensics, a blood sample, a hair sample, a skin sample, any of those will do fine. And that'll also be useful for paternity or maternity if it was needed, testing, because that's exactly what gets passed on to your offspring. But this isn't the case in lamprey. So lamprey has the genome in the cells that give rise to the sperm and the egg cells, so the reproductive cells, is a bit different than the genome that you find all over the rest of the body. And it's different due to a process that's called programmed genome rearrangement. So it's programmed because it's not random. So bits of the DNA just get cut out. And the reason they get cut out, or the probable reason they get cut out, is quite interesting. So you probably know that if you scratch your skin will regrow, it'll heal, but only skin will regrow there, right? So you won't get heart tissue growing there, and you won't get other kinds of tissue growing there, and you won't get liver tissue growing there. And that's because even though the DNA is all there in the cells, a lot of the genome has been shut down. And that's a kind of tricky, complicated process. And if that process of shutting down all the wrong genes in a cell breaks, then things go weird, and one of the weird things that can happen is cancer. So that's when cells are kind of growing out of control and not growing the right type. So this lamprey, one of the things it does is during development, it actually ejects the genes from its genome that it doesn't need anymore. So the ones that are not present in the adult tissues, except in the reproductive cells, are developmental genes. They're those genes that guide the process of growing into an adult shape. They're not needed anymore. And so instead of silencing them the way we do, which is packing tons of proteins and things like that on top of the DNA that just stop it from being expressed, the lamprey has a different strategy, which is just to cut the piece of DNA out and throw it away, which was kind of weird. And it is definitely weird. It only happens in a few small examples of organisms. So anyway, it's a weird thing. That's a total aside, but there you go. That's for free. But if we look at, but the idea is that if we can get the sequence from this genome and compare it to the other vertebrates, we can start understanding that ancestral vertebrate. And so this is what we've done. And so this my collaborator, the guy you could see carrying the elephant shark, he's also sequenced this. So we can look at this and try and figure out what's going on at the base of the vertebrate tree. And so this is what DNA sequence looks like when you print it out. This happens to be a bit of the human genome sequence because the only organization I know that ever bothered to print out the human genome sequence is the Wellcome Trust in London. So they printed out the whole thing and made this enormous bookshelf to carry it all, and only by printing very, very tiny font could they even get it into a reasonable number of volumes. But I'm printing and showing you this here because actually there's this lovely metaphor that has existed for a long time in terms of how we consider our genomes. And that is we can consider our genomes as historical documents. And the first example I know of somebody saying this was from 1946, where this Japanese man, said the history of the Earth is recorded in the layers of its crust. The history of all organisms is inscribed in the chromosomes. So by reading our genome sequence, we are trying to understand about history. And that's what I've been talking about until now. But we actually made this metaphor almost a bit more literal by adopting some methods from document analysis. So, yes, I have these in the wrong order, but I'll go with it anyway. Okay, so what happened in terms of, so I've got some complicated pictures here, which I'm hoping I'm going to be able to explain well enough. So I'll go through them slowly, and so let's see how we do. So in terms of what we are talking about, in terms of what happened in terms of the genome evolution, so imagine a situation that starts here and it just moves across. So we start here, and this simplified version of reality, this ancestral organism, this ancestral vertebrate, just had 3 chromosomes, which we've just colored blue, gray, and black. And when we had the first round, the first 1R of genome duplication, there's just an extra copy of all of these. So that's what's happened here. We've got 2 copies. Sometimes things can mix around a bit, like sticking to each other, but that's okay. And then we have another round of this whole duplication. So this 1 becomes 2, becomes 4, and then we have this little mixy-uppy bit here with things sticking to each other, but it's basically what we see. And so if this was a modern chromosome, set of chromosomes today, for example, a modern set of chromosomes in a human, then we can do this comparison of the DNA sequence in them that I showed you already, where you can see that the letters are the same. So you can recognize the relationships inside the genome. And if we just plot those on this graph here, so if we say here, this is 1 chromosome, and we've got the first gene on this chromosome, for example, if we say where, apart from looking like itself, which of course it does, what else does it look like in the human genome? And we say, well, it looks like the first gene on this chromosome, and it looks like the first gene on that chromosome, and it looks like the first gene on that chromosome. So we put a dot there. And we go to the second gene, we see the same thing, and the third gene, we see the same thing. So we can see that these, when we compare the sequence to itself, we can see that we can see this pattern of how we have these stretches in the genome where they look the same. We can see these duplications. So there's this copy on this chromosome, and there's 3 other copies of this in our genome. So this would be the kind of, if we did this graph just after this genome duplication event occurred, this would be the kind of thing we'd see. We'd see this perfect relationship. There's 1 to 4, and it's all very tidy, and these nice straight diagonal lines, because the first one is similar to the first one, the second one is similar to the second one, and so on. Does that more or less make sense? But then what happens in reality is it doesn't just stay nice and tidy like this. One of the things that happens really easily and readily to DNA sequences is you get what we call inversions, which is just little loops form in the chromosome. And if you imagine in the middle, you get a loop forms and rejoins in the wrong place, so the bits get swapped around in the middle. So if you'd numbered them all 1 to 200, for example, you'd have a bit in the middle where it should have been 50 to 99, and instead it's 99 counting down to 50, and then it gets back to 100 again. So you get these little inversions. So if we did that to this plot, if we take this picture and we let those things happen, so we let the orders rearrange inside a chromosome, then it gets messier. And you see something like this. So instead of it being a straight line there, it's kind of all scrambled. Instead of being a straight line there, this is all scrambled in its own way. But one of the things you can see is that all of these ones that have been shaded pink, they all kind of have that same relationship, though. Even if the lines are all scrambled, these ones are all kind of similar to that, and this and these to each other. And if you look, this little yellow triangle, so we've excluded something being the same as itself, right? That's not interesting. That's too trivial. And inside this yellow triangle, you have none of these bits where you have a copy, because the copies are only across chromosomes, not within a chromosome. So we see 2 things here. So we see this kind of little, this big pinkish triangle with these little yellow segments in it, which are the actual chromosomes not having copies within a chromosome, so only across. And so this is what we expect when we have these steps, these evolutionary steps. And so, and then if chromosomes started crossing over and rearranging with each other, you get something like this instead. So you get these patches, but they seem to be scattered all over the place. And this is the reality. So that's what we have in the human genome. So this is the real data. So this is if you compare the human genome to itself, except looking for copies, you find this kind of scattering all over the genome of you have these patches of genes that are the same set of genes as on some other chromosome somewhere. So we wanted to make sense of this and say, well, is it really the pattern? Is it really the result of these processes that we're talking about? And so the thing that I was trying to say earlier in terms of the document analysis should come now, yes. So we took some models, some methods from document analysis. So making this metaphor literal. And so this is a figure from this paper here. So this guy, Blay, designed these new methods for automatic document analysis. So the idea is that you have documents, perhaps scientific papers, and you want to organize them into topics. So it's a data mining of the, say, for example, a scientific literature. And he designed a new computer science method, or it's a probabilistic method, for automatically assigning documents into topics. And the basic idea is if you have a document like this, it contains all these words, and this method kind of automatically learns that certain topics have a certain collection of words. So if you had a paper that it was about genetics, you tend to find the words gene, DNA, and genetic in that paper a lot of the time. And if you had a paper that was about evolution, you tend to find these words occurring at a high frequency in the paper. And so you have each of these topics has a collection of associated words that are going to occur, kind of characteristic distribution in the papers. So this is what the paper does. So basically, this method takes a document and assigns that document across topics based on the words the document contains. So you can say this document is a bit about genetics, it's a bit about evolution, and it's a bit about neuroscience, this particular document. So this is a method, it's based on probability analysis, and so you get these distributions of words and topics across documents. And so the clever thing that I didn't do, it's a postdoc in my lab who has a primary training in mathematics, realized that he could translate this problem into a genome analysis problem. So instead of assigning documents to topics based on the words they contain, you can change it over and you can assign modern chromosomes to ancient chromosomes based on the genes they contain. So that's the depth of description I'll give you for that. But basically, you have, so there's a distribution of genes, and it's shared. These red bits have similar distributions of genes, so they belong to the same topic, which actually is the same ancestral chromosome. So we can see these are derived from the same ancestral chromosome. And when we do that to that messy picture of the human genome with everything all scattered out, like I showed you before, we can do it and we can retrieve these things that I was showing you before, where you have these pink triangles with the little yellow bits in them. So we can find this pattern that we thought we should find if this history was true. And you can probably just about see that within this big pink triangle, there are 4 of these yellow triangles. So this is one big ancestral chromosome that it has now 4 bits in the modern genome. So we get this 1 to 4 relationship like we expected from this ancient genome duplication pattern. And so now where we can see the relationship of these chromosomes to each other, and we can understand better that these bits are descended from a single segment of chromosome in this ancient vertebrate ancestor. But so by adapting this document analysis, we've been able to successfully infer the structure of the ancestral vertebrate. And that's now, so it means we can see some of these relationships that were cryptic before, that we couldn't identify before. Now we can see them. And part of the reason we're interested in them is because it has quite important implications for how we understand our genome. At least it does for me. I find it exciting, and I'll show you why. So if we talk about human genetic diversity, there's, broadly speaking, two kinds of genetic diversity we can talk about. So between this individual and this individual, or any people I would pick in this room, if I looked at your genome, I'd find that sometimes one of you will have a different letter in the sequence, whereas this individual has a C in the equivalent position, the other individual might have a T. So we have some differences like that, just differences in the letter. But we also get differences in the number of copies of genes. This happens quite easily. So this kind of gene duplication that I was talking about earlier, it happens really, really readily. So if I just surveyed this room, I'd find lots of examples where some of you have one copy of something, somebody else might have 0, and somebody else might have 3 or 4. It's a really, really common kind of thing to happen. And so in many respects, you can say we're all mutants. Every individual is carrying lots of genetic variants, and some of those mutations are totally new to you. So if you sequence a trio, which is a mother, a father, and a child, and you look at the child, you can see that that child is carrying some mutations that it did not inherit directly, from either parent, in the sense that that parent isn't carrying that mutation. It's just a new mutation that occurred maybe in the formation of the sperm or the egg, or maybe very, very early during the growth of that individual. And we all have about 60 of those, which isn't a huge number, perhaps, but if you count up, so each of us has about 60 new mutations. The entire human genome is 3.6 billion letters long, and the population of the Earth is 7-something billion. So you can calculate that every mutation that's viable or that's consistent with life, let's say, ought to exist somewhere on the planet. So not all in the same individual, of course, are scattered, but if you compiled all the mutations from all the people, all those 60s, across everybody, every viable mutation exists somewhere on the planet. So we're all mutants. So that's a good message to take home. But sometimes you can have mutations, of course, that aren't healthy or that can cause conditions. And one way, if we think about just those duplication type mutations, sometimes you can have too much of a good thing. So the gene might be perfectly normal. There might be no change in the sequence. It might be a perfectly good gene sequence that ought to do the right thing, but having the wrong amount of it can cause problems. So if you're supposed to have, in this case, this protein present in 3, whatever our fictional units are, there might be a case where if you have too much of it, that the protein clumps up, and that clumping up can be bad. So this is actually one of the things that happens in some neurodegenerative conditions. So Alzheimer's and Parkinson's are associated with these protein aggregates, so proteins that are clumping up together. And in fact, one of the risk factors for Parkinson's is a particular gene that's normally present in 2 copies, so 1 from each parent. And if you carry this gene in 3 copies instead of 2, so 1 extra copy, there's a higher incidence of Parkinson's in individuals that carry 3 copies. And if somebody carries 4 copies, they might get early onset Parkinson's. So it's just having an extra copy of what is apparently a perfectly normal gene, it increases the chances of this aggregation, which causes the symptoms. And another way in which we might, not only aggregating, but a lot of our proteins work together. So they form complexes. So the hemoglobin was one example, where it was the alpha and the beta together that make hemoglobin. But there's lots and lots of examples like that. So if we imagine we have these 3, this yellow, red, and blue protein that only work when they're together like this. So this is the thing that does the job, whatever that job in the cell is. That's a normal amount. But if you ended up with just extra of the red. So we have yellow, red, and blue, and just get extra of the red. So we have 2 copies of red instead of 1. Then you can imagine a scenario where the yellow has successfully bound the red. This red has bound this blue. And there's no free yellow available to complete this one. And there's no free blue available to complete this one. So even though you've added something, you end up with a deficit, or you end up with, perhaps even with no functioning. So this is another way where you can have too much of a good thing. In this particular case, it's this ratio of the components that's important. So we call this example like a balanced or an unbalanced. So this is dosage balanced with respect to each other. And so if we think about these dosage balanced genes a bit more, so this is just what I showed you there. This is exactly the same one. So if you just do 1 by 1 gene duplication, so this red one happened to get copied randomly, then you can end up with this scenario that could be deleterious. It might be inviable, or it might cause disease or something. But if somehow you manage to duplicate all of them simultaneously, so you have an extra copy of everything, then you're okay, because it's the ratio that matters here, not the exact amount. And if we're talking about these duplications, then we have this way of guaranteeing a balanced duplication. So this is our little Siona friend, which we have this times 4 because the 1 became 2 became 4. So this balance, these dosage balanced genes, if you duplicate them like this, that's okay. But if you duplicate them 1 by 1, once they're duplicated, that's okay. But if you lose them individually, that also isn't okay, because you're going to end up with the imbalance problem just from the other end. So if you duplicate just 1, you get an imbalance. But if you duplicate them all and lose just 1, you've also got an imbalance. So they can get duplicated this way by this whole genome duplication, but not lost again. So earlier on, when we were talking about, lots of these genes get lost again, there's something special about these ones that don't get lost again, because these ones are disproportionately these genes that are very important, that have this very special dosage that really matter. And so when you're talking about these duplications that happen randomly in all of us, and these variations between individuals, like I said, a lot of the time it's completely inconsequential, but sometimes it does cause disease. It can cause developmental conditions, so that you see them already in children, or it can cause later life things like higher risk of heart disease and things like this. And so when we look at the kind of, when we look at the mutations, one way you approach genetics to try and understand what might be a causative gene in these cases is you compare the individuals that have a condition to the individuals that don't. And when you do this, you can see these big duplications that are present in individuals that have a condition that are rare or absent in individuals that don't. But when you look at them, these duplications are really big and they contain lots of genes. So yes, you've narrowed down your search for what's happening, but you might not be all the way yet to really finding out which gene is the problem. And it's important to know which gene is the problem, because if you do want to start to try to develop a therapy for something, you have to understand first what it is that's wrong. What is it you're trying to correct? In one instance, you might need to produce a drug that will take away some excess of some protein. In another case, it might be a different protein that's in excess, or it might be something that's in deficit. So you need to understand what it is before you can really start trying to think about how to fix it. And so in terms of, if we just imagine here, this is a cartoon example, but this is, imagine a stretch of a human chromosome, and these boxes on it are supposed to be genes on the thing. And so we have these little lines that are above and below are just representing these extra copies, these different number of copies. So we call them CNVs because they're copy number variants. I should have taken that jargon out of the slide, so sorry about that. But it's just a variation in the number of copies. That's what that is, copy number variant. And I've shaded them gray when they're present in individuals that don't have any condition. So you can see lots of these are happening and they don't matter at all. But sometimes you find these copies, these extra different number of copies in individuals that have a condition. And so you want to try and zero in on this and try and say, well, what's happening in these individuals? Which genes are causing the condition? And so you can kind of do a first pass, which is excluding. We say, well, if these genes have changed copy number in some of these healthy individuals, we don't think it's those. And we don't think it's those because those can also change in healthy individuals. So you narrow down into this bit because this is the only bit that you find copied and associated with condition. And so here I've shown it with 3 genes in this region. Sometimes it's 40 or 50. And so then what we can say is we can bring in this evolutionary information about the genes that we know by the way they evolve, the pattern of evolution looks like the way that a dosage-sensitive gene evolves, because what we're seeing here is just a change in dose of the gene. We get an extra copy. And so then we can bring in this evolutionary information to narrow in on the genes that are possibly causing these conditions. And when we do this, it looks like it works really well. So we take this kind of information from human populations. So we have these examples that are benign, there's no condition associated, these examples that are pathogenic. And if you just, to test the idea, you just ask, well, these genes that have this special pattern of evolution, these genes that evolve as if they are dosage sensitive, where do you find them in the genome? And you disproportionately find them in these parts of the genome that are associated with disease. So it looks like the approach works. So then you can take it a step further and try and use it in the cases where you don't have that information. So that's what we do. So hopefully what I have explained in an intelligible way is that we use this strategy of comparing the genomes of the different organisms, just like you can comparing their physical traits. You can compare their genomes and make inferences about that ancestral genome that you can't actually see. And so we can take this document analysis method and infer our ancestral genome, and we can learn about these genome duplications. And we can also figure out that because of the dosage constraints, so having to have the right number, the like Goldilocks, just right kind of number of copies of a gene, we can see these characteristic patterns of evolution of some genes, and we can relate these to human disease, and they're quite informative in the discovery of human disease genes. So I'll just say thank you to the members of my team, especially Yoichiro. So he's the mathematical genius who was able to do the document analysis stuff. And this is Venki, who sequenced the lamprey and the elephant shark genome. And I'm very happy to take questions if you have some.
Well, absolutely fascinating, and how fascinating that exploring the science of what actually goes on brings you back to a position where you can understand things, where we can cure people.
Yeah.
And that's just wonderful. Now, the arrangements for asking questions, do we have a... We do, yes. So as the regulars will know, rather than me trying to remember who's put their hand up, I have two splendid assistants on the left and right, and the trick is to catch their eye, and I shall go from side to side. So catch their eye, they will bring you the microphone, and then if I feel like it, I'll point to you, and you can have a go. Would we like to start up on the left-hand side this time? Anybody on the left-hand side? If not, your chance has just gone, and I know there's somebody on the right-hand side. Here, please. Sorry, on the right-hand side first. You'll have to be quick.
Oh, I'm the right, not the left.
Yep.
Okay.
It depends which way. Yeah, that's what you mean. Yeah, sorry.
Okay. Thank you very much. In your description of this process, you described the, I think, computer program that does the analysis as a probabilistic program. And also you said the genes have definitions or can be defined in different ways.
Sorry, the genes have, say again?
You said a gene was defined in different ways.
Yes.
So there's fuzziness about the gene and fuzziness about the way you extract that information. So I wondered, when you go back and look and think that you've identified an ancestral gene, what sort of accuracy do you think you have?
Okay, so genes there is a fuzziness in the definition of a gene, but we just use one definition for this. So we're only using one at a time. So that's one element of fuzziness, I suppose, that is removed. So there's certain examples that are quite easy to be confident about because they're so strikingly similar. So, and they're the ones that we use for the analysis. So when you look at the sequence, and so they're very long sequences, and you just look at the letters, they just match really, really well. So the most extreme examples are extraordinary. So the best example I know of is a gene that between human and wheat So where the common ancestor is about 1 billion years, I'm not sure. Nobody's 100% sure, but really ancient. And whatever that organism was very simple. But there are only 2 differences in the protein between human and wheat. So that's really obvious. They're not all so easy. But the ones that are really hard and ambiguous, we tend to exclude from the analysis in order not to corrupt the analysis. But there's enough of the easy ones left over. But the probabilistic method, the thing that gives us confidence in it, I didn't show the data here, but when we compare the results of that across outside to the things like the C-squirt and other things out there, we see very distinctive patterns. So they're not distinctive patterns of the letters matching, but distinctive patterns of the clustering of the genes in our inferred ancestral genome with the actual genome in those things, and there's a very good correspondence. So it looks good.
Good evening. Thank you for the talk. I was wondering, is the process of whole genome duplication, is that something that's still ongoing? So could feasibly someone in this room or someone alive have their whole genome duplicated, or did it cease at some level of complexity?
So it is, it's not ongoing as far as we know in any organisms that have chromosomal-based sex determination. So because it seems to screw up the process of development. So the event can still happen, but it's associated with spontaneous terminations. So in humans, you can see sometimes if you look at spontaneous terminations, when they investigate them, you can see it. But for example, it happens readily still in fish, and salmon has a very recent genome duplication, which its genome is still in the process of stabilizing afterwards. So when you have the genome duplication, you have 4 copies of everything, and you have 2 copies of all your chromosomes, 1 from each of your parents. And that's a more stable, having just 2 copies is more stable than having 4. So every ancient example we know where they have these extra copies has reestablished just this pairwise chromosome system. And in salmon, it hasn't completed the process yet. So the chromosomes are still, some of them are kind of in pairs again, some of them are still in 4s. So the process happens, and it happens really, really readily in plants as well. And so wheat, again, that I mentioned, has 6 copies of everything. So there's been hybridizations between different wild wheats that have given us our domesticated wheat. And then the brassicas are all different examples of these genome duplications. So we got whichever brassica you're looking at. So cauliflower and broccoli are different genome duplication set than things like oilseed rape and some of the other brassicas. So it's an ongoing process. And it's quite common in one sense. The thing that is rare about it in the context of animals is whether it leads to a productive evolutionary line or not. So in our history, it's happened twice. In fish, it's happened some extra times, but you can still count them on one hand, So.
Hello, thank you for a very interesting talk. You mentioned at the end the association of gene configurations with different diseases, Parkinson's and heart disease you mentioned. The nature of research groups, I guess, is that they tend to study a disease and try to find something wrong with the disease. Do you think it's ever going to be possible? Are we approaching the point where a doctor will take a sample of your tissue, run the genome and say, aha, you have not just a risk of heart disease or a risk of Parkinson's, but you have this and this and this and this. Is that feasible?
So yes and no. So these statistics, they kind of they make sense for a group, but not an individual in one respect. So if I was to say, if I have 1,000 people carrying this particular mutation, I could say, I could be confident that on average, more of them will develop a condition than individuals that don't. But for a specific individual, the predictive value is not so high. But so you can you can tell that person they have maybe a slightly elevated risk, but you wouldn't be able to tell them necessarily that they will or will not get something. But as an interesting, it wasn't what you asked, but what I thought you were going to ask in the beginning, but there is something that now happens in terms of, instead of starting from the point of view of a disease, there's more and more examples of where people have a diagnosis without a name, and so they have some symptoms, not a diagnosis, some symptoms without a diagnosis. And so these tend to be incredibly rare diseases, so they haven't really been described. And there've been examples now, a few examples that I know of, where they have an individual with a set of symptoms and no diagnosis, and they've taken a genetic approach and discovered a mutation that appears to explain the symptoms. And so they start getting towards an fundamental understanding of what's going on in this individual. So they start learning then what it is they need to correct in order to alleviate symptoms, in order to develop therapy. So it is coming around the other way. So you start, instead of saying, I'm an expert in this disease, I collect samples from people with this disease and try and understand it. It's sometimes coming the other way when there's symptoms and no diagnosis.
If I can come back to the...
Oh yeah, sorry, I was just looking in the wrong direction. I didn't know where to look.
I thought I might have been talking in the wrong direction. So if I come back to the previous question, is this process of duplication a feature of organisms with sexual reproduction, and is it related to the meiotic process?
So with sexual reproduction, yes, but not with sex chromosomes. So fish, like I said, fish have this, there's new examples of this happening in fish, but the only example I've ever heard of in a mammal was a South American rodent where it had an extra set of copy of every chromosome except the sex chromosomes. So it was almost the exception that proved the rule, and that it kind of made it look like, yeah, there's really something about the sex chromosomes that have got to be just in the right ratio for development to occur properly.
I hope you don't mind, but rather than asking a question, I'd like to comment on your presentation.
Okay.
My specialty is in the teaching methods which are most highly effective.
Okay.
And in terms of combination of words and images, I would give you at least 9.5 out of 10.
Oh, thank you.
One of the mistakes that many presenters make is to have very complicated slides, and I think you've done an absolutely wonderful job in making it as simple as possible. I don't know, you know the adage, as simple as possible, but no simpler.
Yeah.
And I also thoroughly appreciated the way in your verbal presentation you removed as much technical language as possible. And I think you've managed to communicate to us an immensely complicated thing in a very simple way. Thank you very much.
Thank you. Thank you very much.
Absolutely fascinating.
Thank you.
I'm an engineer, so I don't understand very much of this at all. But can you, from the statistics and the mathematics, tell us what will happen in the future? This is evolution. Have you already started to predict? I mean, we've got all sorts of interesting fictional ideas of how we might develop in the future, and in which direction are we going? Are we going to lose certain features because of technology, or is this beginning to occur? Let's put it that way.
So kind of yes and no. If I imagined a very specific set of circumstances, I could then predict under those conditions certain things. So one thing that we do see in relatively recent human evolution is change. We have drastically changed our relationship with the pathogens around us by our behavior. So a few things we've done, so we cook food, we have hygiene practices, which mean that fewer bacteria and other pathogens get into our body. And this has changed how our species is evolving, because one thing that's happened is we have lost lots of immune genes in a good way, actually. It probably doesn't sound like it immediately. But any immune system gene has its good part, which is that it can recognize a pathogen and destroy it. But it also has a cost, in a sense, which is autoimmunity and things like septic shock and these kind of things. So these are the immune genes that are going wrong. And so if you imagine it like that, you can think of every gene having kind of this cost-benefit balance. And if you have the cost of a gene, which is some low risk of sepsis, and the benefit of a gene, which is eliminating this pathogen that we meet regularly, then the benefit far outweighs the cost, and natural selection will mean we retain this gene in our genome. But if we then do something in our behavior that eliminates that pathogen, almost eliminates the incidence of that pathogen entering our body, that benefit is gone because it's no longer protecting us, we're protecting ourselves another way, and all you're left with is the cost. And so those genes, some of those genes have been eliminated from our genome. So we can see these patterns. So under precise circumstances for specific genes, we can talk about things that, yes, if we really don't use them anymore, then there's no mechanism that keeps them in place. So the natural selection, most of the time, natural selection is keeping things there rather than elaborating them and making them new and fancy. We don't think about it as evolution, but the biggest thing that's happening in evolution most of the time is keeping things steady. So it's rejecting bad things, screwy things that have happened. So you can make predictions under specific circumstances, yes. But the no part is, if we rewound the clock back 500 million years to those genome duplications at the base of the vertebrate tree, there's no guarantee we would have evolved at all. So you can't, in a certain way, you can't. So it's there's so much chance involved, and it's so complex that even if we were to rewind and let it run again, we wouldn't necessarily get the same outcome. So. Yeah, that's why engineers probably are happier with systems that they can predict.
Professor, first of all, congratulations for the critical way of thinking and for your historic, I should say, discovery.
Thank you.
I would like to ask you if you believe that the disease-sensitive genes are connected with an evolution pattern in any case.
Sorry, I missed a word. Our what pattern?
With this evolution pattern you described, in any case, if you have a mechanism describing the timeline, the causes, and the of these evolution changes in the genome.
I'm sorry, I didn't fully understand your question. Could you maybe rephrase it?
If you have a timeline and a mechanism describing these changes you described in the evolution in the genome, and if you believe that the disease-sensitive genes are connected in any case with this pattern.
Okay, I'll try and answer. I see if I understood you correctly. So, okay, so these genes, so my understanding is that these genes are, so they're dosage sensitive and they are linked with disease because they're very, very important when they work correctly, and if you disrupt them, it's disrupting a very important process. So these genes are important anyway, and the duplications and the various things that happened in our history didn't make them more important or change their importance. It just gave us a device, in a sense, to be able to recognize them. Because if we look at these genes in other, more distant organisms that didn't experience these genome duplications, we can still see patterns there that suggest that they are extremely important genes that cannot change. They cannot accept evolutionary change without consequences. Is that answering your question? I'm not 100% sure I understood your question.
Are there any genome diseases who are not connected with
Okay.
a pattern.
Yeah, okay, so this doesn't identify everything, is that's what you're asking. So yes, so this is, there's more to it than just this. There will still be other, dosage sensitive genes, and there'll be other genes that are associated with human disease for other types of mechanism and other types of reason that won't have this pattern. So it's not the only pattern of evolution for a disease gene, it's just one of them. Did I hit this time? Okay, great.
Brilliant, thank you.
Where does Down syndrome fit into this mix-up?
Who asked me that? Okay, actually, it's interesting that you asked that because I didn't put in the slide on chromosome 21, but I had one. So, Down syndrome is caused by an extra copy of chromosome 21, which is, so it's a bigger example of this, too much of a good thing. And actually, when we look at the genes on chromosome 21, we see that so first of all, there are some genes that have already been described as being associated with the syndrome. So not every gene on that chromosome is causing the syndrome. Some of them are causing the syndrome. Some genes have already been associated with that. And if we look at those genes, nearly all of them have this pattern of evolution, this that I've described, this retained after the genome duplication. And there are some more that weren't previously associated with Down syndrome. And we think that those ones are also interesting for further studies. So chromosome 21, so the reason we seek Down syndrome is because having an extra copy of chromosome 21, it does cause symptoms. So we have the syndrome, but it isn't, but they're it's quite a mild phenotype. It's quite a mild condition. So we have 22 chromosomes plus the X and the Y. And any of those 22, you can potentially have the event that you have an extra copy of the chromosome. In terms of when the egg and sperm fuse, you can have any of those 22 might have an extra copy. But we see Down syndrome in about 1 in 500, 1 in 800 live births, not because chromosome 21 has an extra copy more than others, but because it survives better, because the other ones are incompatible with life. And consistent with that observation, chromosome 21 has the smallest number of genes that have this pattern of evolution, which is consistent with having the smallest disruption in terms of the growth of the organism. The next most common trisomy is trisomy 18. And trisomy 18 has the next smallest number of these genes. And the next most common trisomy is trisomy 13, which has the next most common. So the smallest number, the second smallest number, the third smallest number are the most common trisomy, the second most common trisomy, and the third most common trisomy. So it looks like these genes are connected with the trisomy in terms of the symptoms and also the viability and the fact that it's quite a mild syndrome. Yeah, that answer?
I'm no biologist, so you must be gentle with me, but current approach seems to be to identify a symptom and then look for a genomic explanation. Is anyone working the other way and making the changes in the genome and then looking for symptoms?
Making the changes?
Yeah, you could chop them around. You know, swatch them around.
So not in humans, but that's actually the classical genetics approach to create mutants. So the classical genetics approach is to make mutants and see what happens. So there is kind of one of the early labs that ever worked on genetics was called the fly rooms in Columbia University because they worked on fruit flies, Drosophila. Okay, so to make mutants, yeah, so it's still an approach that can be used is to make mutants and to see what happens. So one of them is that you, the kind of extreme version is you remove a gene entirely and see what happens. And as I was telling some of my hosts earlier today, the surprising thing is that even though some of the times this might be a gene that we are sure is really a gene based on this pattern of conservation and we can see it in all these other species, sometimes nothing happens even when you remove the gene, which tells you that evolution is sensitive to tiny, tiny variations that we can't even observe. But yes, also other times in a model organism or in a cell line. So the experimentation that is done on humans in a sense is on human cell lines. You can have cell lines to grow in a dish in a lab. That won't tell you about complex functions, but you can look at the functioning of a cell, and so you can deliberately change a gene and see what occurs. So that approach is used, yes.
I think Dr. Jekyll and Mr. Hyde had a go at this, but it wasn't altogether a good outcome. So cool. Yeah, over here, please.
Thank you very much for that. I'm fascinated by this idea of dosage-based pathology.
Yep.
So if I'm understanding this correctly, if you have surplus copies of a particular gene, this leads to the sort of pathological change.
Sometimes, yeah.
How can you end up then with a treatment pathway that will deactivate...
With a what pathway, sorry?
A treatment pathway
that
will deactivate sufficient of the copies that you return to the optimum number.
It would really depend on a case-by-case basis of where in the whole system that disruption has occurred. So for example, sometimes the disruption occurs during the process of development, which is growing from that single fertilized egg into a little newborn baby. And in that case, you only notice it after the fact. And so there's no going back, in a sense. Other times, if it was, say, for example, like the risk factors for Parkinson's, that's a late life condition. So there is definitely a therapeutic window and time for intervention, but it's not necessarily easy to get the therapy there. It's notoriously difficult to get anything into the brain, even standard medicines. So it depends on the case of what is. So it's going to depend on a case-by-case basis of what's happening, how accessible the tissue is, if it's a whole body thing, or if it's just some tissues that are affected. But yeah, so sometimes it might still be that therapeutic option is not trivial. Well, it's never going to be trivial, but even less trivial.
Thank you. And there's a question on the right here.
Hello, thank you very much for your talk. After the question about where does Down syndrome fit into this, I was thinking about mosaicism, because I know you can get some genetic diseases where you have a copy of a chromosome, but only in certain cells. Has that ever been shown to be beneficial, and how does that, I suppose, happen?
So how it would happen is more than likely a spontaneous mutation during the process of development and growth. So, for example, you will sometimes see individuals and their two eyes aren't the same color as each other. And that can be a little mutation that occurred during development. And so you have a mutation in one eye or the other, or maybe one side of the face or the other, and you have this physical manifestation. So you can get mutations that occur during development that mean that not every cell in your body has exactly the same DNA sequence. So getting an extra copy of a chromosome could be another example of that. And so some cases of Down syndrome that are very mild are due to mosaicism. And so it could either be that during the process of development, some cells and therefore all their descendants, let's say it happened at a 4-cell stage in a growing embryo, then if one of the cells manages to somehow not receive somehow eject the extra copy of chromosome 21 by that mutation, however it might happen, then roughly a quarter of the body is going to be have a normal karyotype, and normal number of chromosomes, and the other would have the extra one. And so you can get these mutations that occur as you grow, which mean that you have slightly different gene sequence, DNA sequence in different parts of your body. And in fact, if you took a sequence from your right hand and your left hand and you sequenced the whole thing, you would find differences. But most of the time they're so slight. Well, they happen in the 98% of the genome that doesn't really contain genes, and so.
I think we must let Professor McLysaght off the hook in a minute, but there's one question here and a final one down at the front here.
So what I learned as a student about 1 million years ago is that the thing that puts the sea squirts and us in the same phylum, the chordata, is the presence of the notochord during development. So can you identify genes that are associated with the notochord, and then can you find related genes in things that are not in the chordata?
I bet someone could, but I've never, I haven't tried to do that, so I don't know. It sounds like something, at least it would be plausible to try, but I don't know if it's been done. So.
And the final question at the front, please.
Well, thank you so much for a really interesting talk. I probably haven't understood it properly, but my question was, in all of the developing and the copying and the progression, is the rate of speciation carrying on in the same way?
No.
Is there anything can check it naturally within the system?
Can check it. Right?
Check it. I mean, can you go on copying and having stuff around a new species indefinitely? And is that going on at the same rate as in the early stages?
So we do get spurts of speciation, and actually after the genome duplications, there probably was a radiation of lots of different species as they resolved that instability in slightly different ways. And so these genome duplications are probably associated with bursts of speciation, but speciation is ongoing as far as we know. It's not the kind of thing you notice in real time, but most of the species that have ever lived have gone extinct, like something like 99 point something probably of the species that have ever lived have gone extinct. We see the ones that are around today, but we expect that these processes are ongoing. The only example I know in a vertebrate where anybody has noticed speciation occurring was in the Galapagos. A new species of finch was observed in the last few years. And that is partly down to the extraordinary work of two people who've been studying those finches since, I'm not sure if it's the 70s or earlier, but it happens to be a married couple who go there every summer and camp on one of the islands. And I think they know each bird individually. They see them nesting, they watch them hatching, they observe them, and they noticed a new bird. And nobody else would have been capable of noticing. But then with some more follow-up, it was realized as well that this was a new species. It was a hybridization, I think, between birds from different islands, but it established a new species. So it's rare to catch it in action, but we presume it's ongoing.
Well, it's a fascinating note to end on. Professor McLysaght, it's been absolutely fascinating.
Thank you.
I think the gentleman halfway up stole my thunder in saying what an exemplary presentation it was. Thank you for that. And also you've taken us from the most ancient creatures to the fact that things are, new creatures are being created. And also, I think, bringing to bear what we would perhaps call big data.
Yes.
This probabilistic analysis of what's going on, which guides us towards mechanisms so we can understand how they work. Could hardly be more fascinating. And now when I go home and explain to my lady wife that I started off as a piece of tubular intestine and a bit of wheat, she'll say, well, I don't think you've evolved much. But really, a fascinating evening. Thank you very much indeed.
Thank you. It's a pleasure. Thank you.